Hello students, welcome you all. So in this session we are going to start taxonomical aids. In the previous one we have done about the taxonomical categories. Fine. So what is the meaning of aids? Aid means help. It means help which is we are taking. So what type of help? For taxonomical studies, what are the things we are taking help? For example, different procedures, different techniques for what? To store, preserve, to store, preserve and the information and the specimen. Fine. We know that because for example, if I am doing any research today, so maybe tomorrow I will not be there. Fine. So what will happen? My coming generation can study or can do my work for the further studies with the help of these aids. What we are doing? We are preserving our information. Whatever the studies or whatever the research I have done, it will be done by the other generation, by the another generation. So for that one we have to preserve all the information and all the specimens. So taxonomical aids are the help which we are using for the taxonomic categories or for doing the taxonomical work. And what is taxonomy? Categorization. Clear? So here now we are having different aids, different devices we can say. The first one is herbarium. We all know about the herbarium. In schools what we are doing? We are drying the leaves or any sample of the plant and they are pressed into below the some heavy object or we can say in the books, or after that what we are doing? We are preserving them. So dried, pressed and preserved, and where we are doing keeping that in the sheet, in the paper sheet, a thick sheet. So here if I have pasted one sample of any leaf showing any characteristics, for example simple leaf, compound leaf, or any inflorescence or any root structure, tap root and fibrous root. So we can show any of the characteristics with the help of this sample. Fine. So as you have pasted it here, now what you will do? You will provide some information when you have collected this leaf and who has collected this leaf, what is the name of the plant, what is the scientific name, what is the common name. So you have to give all the information here. Fine. So after that you have made different types of sheets and arranged accordingly the rules and regulations. So in herbarium what we are doing? We are taking some plant or a sample from the surroundings and we are keeping in a sheet of paper and we are preserving it for the future use. For why? For the future use. By this what will happen? The sample, if we are getting any sample, okay, so what we will do? We will compare with the existing one. So what are the characteristics? They are matching with the existing one. So these are the important things. Second one is museum. We know about the museum. It can be in school also, in colleges also, in any town. So this museum is what? It's a preserved plants and animal specimens. So what we are doing? We are preserving some plants which are important for us or which we have collected from any important place, and along with this we are dealing with the animal species. Memorize one thing, whether it is a herbarium or museum, we are talking about the non-living or not the non-living, they are not about the living part. Fine. Here what we are doing? After dying of plant we are preserving it, and after dying of any plant or after dying of any animals, then we are preserving them. But in other cases, in case of botanical garden, in case of zoological park, what we are doing? We are not dealing with the dead one, we are dealing with the live one. Fine. So in museums again we are taking those plants and animals which are not present now and we are preserving them. So there are different methods to preserve them. For example, sometimes what we are doing in case of plants, we have collected some plants and any specimen and we have kept at one place. Another method is that preservation. In your schools you have seen that some specimen or some animals, for starfish, you have preserved in a solution, formaldehyde solution. What you are doing? You have kept the starfish. So what is happening? You have preserved in a jar or in any bottle in chemical. Another method of preservation which you can see in the museums also. What are these methods? For example, in case of mammals, in case of big animals, what you will do, you will take out all the inner part of the animal and you will go for the stuffing of any material or form you are stuffing in that organism. For example, it is any of the animal and you have stuffed this one and you have kept it in the museum so that the coming generation can see that what it should be. For example, in case of dinosaur, any fossils which we are getting, what we are doing, we are preserving in the museums. As well as what we are doing, another method of preservation is what? For example, we are having an insect, we are having butterfly. Fine. So how we first we will dry this butterfly and we will put it into the jar. Okay, so we will put into the insect box or the glass box, and what we will do, we will go for the pinning. With the help of pins, we will fix them into this box. This is another method. So there are different methods to preserve these plants as well as animal species in the museum, but after dying, not before the dying. After that, the third aid is botanical gardens. What are the botanical gardens? What we related to water? Something related to plants. So in this garden, what we are doing, we are growing, we are breeding, we are cultivating the plants so that we can compare with the other one. For example, here I have written collection of living plants for the reference. What is the meaning of reference? So that if you will get any plant or any tree, so what we will do, we will compare with the plants which are in this botanical garden, so that we will come to know that yes, it is just similar like to that that is present in the botanical garden. So here there are some examples which is very important. Okay, memorize it. What are the different botanical gardens? Kew is one of the botanical garden which is present where? In England. And another one is Indian Botanical Garden which is situated in Howrah, India. And NBRI, National Botanical Research Institute in Lucknow. So these are the botanical gardens. Here what we are doing, we are keeping all these things in a living form. We are not killing them or we are not taking the dead objects or dead organisms. Now the fourth one is zoological park. Zoo related to what? Something related to animals. Here related to plant, here related to animals. So now what we are doing, we are taking animals from their natural habitat and keeping in a protected environment. We all know about the zoo. What are the zoo? In zoo you can see different types of animals are there. In these different types of animals, what happens? We are giving shelter to them, we are giving food to them. Everything is done by the human beings. Okay, so this is the protective environment condition which is done by the human or the under human care. So these are the important things about the taxonomical aids. Another one is the key. Now key is what? It's another taxonomical aid which is used for the identification of plants and animals. How we are classifying? On the basis of their similarities and dissimilarities. How they are similar to each other and how they are different with each other. Fine. So on this basis we are using key. And key is what? It is not like that key. It is some information is given into these keys, and on the basis of these things what we are doing, we are selecting. It's just like a puzzle. How we have to do? It's based on the contrasting. What is the meaning of contrasting? Different characters. If you see this key, this one is a question. This one is the first key or the question. So we are having two types of questions. Okay, in each key what will happen? There will be two different contrasting characters or two different contrasting options, and you have to choose any one. So here what will happen? There are what written couplet. What is the meaning of couplet? Couplet are the pairs. For example, in this key, two things are given here. Organism is one cell or organism is multicellular. So what is happening? These are different characters. It is a one cell character, it is a multicellular character, and both of these two things, it will form what? It's a couplet. What you will say? These are couplet. And again, in each key, in every key I have shown two different character. Nucleus present, nucleus absent. Both are contrasting and they will form couplet. Fine. Again, in the third key, two pairs are there. Both are the contrasting one. Again, they are the couplet. Again, in this key also couplet. So in every key you will find that couplet is there or the pair of contrasting character. Now in each thing, for example, you got one sample and you have to find that in which classification it will come. So how you will do all these things? First of all, you will start from the first character. You will match the sample and you will find that whether this organism which you are having, it is one celled or multicellular. It means what is? It is unicellular or multicellular. So if you will see, so these organism which we are talking about a cellular level, you can see only in the microscope. So if it is a one cellular, what you will do? It will say you that go to the key number 2, right? And if you are selecting that organism is multicellular, if you are observing the characteristics of the multicellular organism, then you have to go for the key 3. Now I am assuming that the first condition, the organism is one cell. So the answer which I am getting here, they are saying that go to the key number 2. And what, see, every key 2, key 3, these are the leads. What you will say? Lead. In each statement you will find some lead, and following this lead you will get your answer. So if you are selecting the key number 2, it will be the lead, and if you are selecting the key number 3, it will also the lead, because on the basis of these leads you will get your conclusion. So we have selected the first one, organism is one cell, so we have to go where? Into the key 2, and if you are selecting key 3, it is a different condition. Now we are moving towards the key number 2. Again in key 2 we are having two different characters, two contrasting characters, those are couplet, right? So if we are getting the answer key in another key, you will find another option, nucleus is present or nucleus is absent. So if you are saying one cell, in U we are talking about the unicellular. So now what you are observing, in this unicellular, nucleus is there or nucleus is absent? So if you are finding that nucleus is present, now you got the answer, that is, it may be the protista. What is it? It may be the protista, because we know that protista is what? It is a unicellular eukaryote, which you will study in the next chapter about the all characteristics of prokaryote, eukaryote, monera, protista, fungi, plantae, animalia. Right? So you will study all about the characters. Here what you have to do, on the basis of some character, on the basis of some lead, you are getting your answer. So you got your answer, protista. Fine. Now if you say that you are finding that nucleus is absent in that unicellular one, you will go for the next one. Next one is also a lead. From this lead you are getting answer protista. If it is not there, if nucleus is you are finding that nucleus is absent, so what should be your conclusion? Maybe it will be the monera. Why? Because monera are the prokaryotes, and what are the prokaryotes? Those who are not having their proper nucleus or having only the not a proper distinct nucleus. So this is a feature of monera. Fine. So you are getting your answer, monera and protista, when you are selecting the organism is one cell from the key 2. Now if I say that this sample which I have taken, it's a multicellular. Now it is not a one cell, it is a multicellular. So the lead which you are getting here, key number 3. So which lead you are getting here? Key number 3. So you have to go in the key 3. Again in the key 3 you are having contrasting character or the couplet. So the first one is the autotrophic and another one is the heterotrophic. Fine. So you have to select what you are finding, autotrophic or heterotrophic. We know that what is the characteristics of autotrophic? Those who can prepare their own food. So if you are getting any idea about the sample, that whether it is a autotrophic, so what you will say? Answer is plantae or the plants. All the plantae kingdom or the plants will come under what? Under the autotrophic and multicellular, because these are the characteristics of plants. In case if you are getting that this multicellular organism is not autotrophic, so you will go for which lead? For the heterotrophic. Fine. So when you are choosing this heterotrophic, it will say to you that go to the key number 4. If you have selected autotrophic, then you got your answer. But if you are observing the characters like heterotrophic, so you will get another lead, which is said that you have to go in the key number 4. Again, when you will go in the key number 4, you will find two pairs. Again, two contrasting character. What are they? Motile and non-motile. Now, what is the meaning of motile? Those can move from one place to another place. And what are the non-motile? Those cannot move from one place to another place. Now, again, if you are observing that it is motile, you will get your answer animal. It means that the sample which you are having, it is somehow animal. And if you are getting any observation that it is non-motile, you will get your answer fungi. Fine. So motile is what? From here, if I will go in the reverse, so what you are observing? Motile animals. Animals are heterotrophic mode of nutrition. Those who cannot prepare their own food, they are dependent on other organism. And this heterotrophic is what? Multicellular organisms. Fine. Now, if you talk about the fungi is non-motile, and if it is non-motile, again it will come under heterotrophic because they cannot synthesize their own food. They are dependent on the dead and decaying part. So, again this heterotrophic is a multicellular. Right? So, like this we have compared all the things. On the basis we will get our answer, and in each of the key, if you are observing, in each key you are getting lead, and this lead will takes you in the another key, and finally you are getting your answer. Fine. So this is about the key, which is very important. Now, along with this these aids, some flora, manual and monograph. These are the different flora. In flora, what happens? Flora means related to plants. So it's a book in which there will be a description of all the plants of particular area. For example, this is an area, all the plants which are present in this particular area, we will study about it, and we are representing, or we are giving description in a book. Now, what is manual? Manual is one of the taxonomical aid, in which what we are going to do, in which we are studying about the define. For example, here we have done only about the plants, but here we will study about all the species in this particular area. Here only we are studying about the flora, only plants. Here we are studying about the all species. The species, it can be plant species, animal species, microorganism. So we will study all the species in that particular area, and we are presenting in a book form, or this is known as manual. Now, what is monograph? Monograph, when we are studying or we are giving description about one taxon. For example, we have done different taxon, kingdom, phylum, class, order, family, genus, species. So if we in this monograph, what we are doing, we are giving description of any one taxon. What we are doing, we are giving description of any one taxon. Well, it may be species, then all about the species. If it will be a taxon, kingdom, then we will give all the description about the kingdom. Fine. So this is the monograph. These taxonomical aids are very important for the classification, because with the help of these things we can preserve our material, as well as we can preserve our information for our generation, so that they can take advantage of these things. Fine. So for today that much is fine, and thank you.